Hi, I just wanted to hit on a few things in Learning Unit 1 before you all take your test. There's just a few things that when I look back over the assignments that I thought might need a little more clarification, and just some things, looking back over the readings, I thought that I might wanted to hit on a little bit more. This is by no means an exhaustive study tool for the test, so you're certainly going to need to study and everything, but these are just a couple of the high points that I wanted to hit on. There is also a section that is your learning objectives for Unit 1. Check those out as you're studying, because those are things that it's expected that you would know by the end of the unit. So check out those learning objectives as you're studying. And also too, when you submit your assignments, once they're graded, go in and check your feedback. I leave feedback for individual questions. If you miss a question, if points are taken off, you'll always get feedback. Sometimes even if you get a question right, you'll still get feedback. Just keep check on that as well. But a couple of things about Learning Unit 1, and I have a little cheat sheet in front of me so that I won't, so that A, I'll stay on script a little bit, and B, I won't forget something that I wanted to mention. But we're dealing with pathogens, specifically foodborne pathogens. And we start off talking about the prokaryotes, which are the bacteria. And the prokaryotes are going to be your simple guys. And we talk about gram-positive and gram-negative bacteria. Now, one thing that that I want you all to be sure that you remember is that the gram in gram-negative is always capitalized. And I know that sounds just so nitpicky, but it's actually the last name of the scientist who invented the gram stain. So gram's always going to be capitalized whenever you write it. And whenever you're writing the name of an organism, that genus and species name, the genus is always going to be capital. The first letter is always going to be capitalized. The species is lowercase, but when you type them out, you'll italicize them. So just don't forget to do that when you're writing them. So we talk about gram-positive, gram-negative bacteria. Gram-positive are going to have that thick peptidoglycan layer. Gram-negative are going to have that thinner peptidoglycan, but they're also going to have that LPS layer, or that lipopolysaccharide layer. That's going to contain lipid A, which is going to be the endotoxin. So, be familiar with their differences between gram-positive and gram-negative bacteria. When we talk about prokaryotes, we also talk about binary fission. And binary fission is just that process of one cell replicating everything about itself and dividing to make two cells. So that's what happens with binary fission. And binary fission, the result of it is going to be exponential or logarithmic growth. And what that means is with every replication cycle, the number of cells is going to double. So you'll go from one cell to two, and then two cells to four, four to eight, and so on and so forth. And the amount of time that it takes that entire population to reproduce and essentially double is the generation time. It's also called the doubling time. So in that time period, that colony of cells can double. They'll go through binary fission, and the total number will be double what it was before. Really important, because if you think about it, once the first couple of hours, it doesn't look that bad, but when you start calculating it out with generation times, you can get to really high numbers of these guys really fast. So that's definitely of concern. Another thing that was covered in the unit, we talked about normal flora, and we talked about opportunistic pathogens. And your body has billions of normal flora in and on the body. There are some normal flora that when conditions become favorable to them, which we would think unfavorable to us, but when conditions would become favorable to them, that they can grow and thrive and become what we call opportunistic pathogens. And those conditions can be things like you can have, they can be introduced to a different part of the body. So we see that a lot with your fecal-oral transmissions, and you get sick that way. You can have a suppression of the immune system, and these guys can flare up, or you can have a decrease in your normal flora that are typically there. And we see that a lot with antibiotic use. Sometimes, antibiotics can take out the good guys and the bad guys. And so when these normal flora are killed off, parts of your normal flora that are still there can grow unchecked, and they can grow into higher numbers where you don't want them to be. And a lot of times you can see that a good example of that is oftentimes there can be yeast infections that happen with heavy antibiotic use. And it's because that normal flora can get killed off, and then it'll allow that yeast, who's typically there in lower numbers, to grow in higher numbers. Infections and intoxications. I think on the assignment, most everybody got a good grasp of that. But for an infection, what happens is the bacteria have to get in the body and grow, and they have to grow to a high enough number to cause an illness. With intoxication, you're consuming something that's already going to have the toxin present. So you're going to get really sick really fast with an intoxication. And a lot of times that happens with Staph aureus. Staph aureus produces that pretty potent toxin, and you can get sick from him quite rapidly. And so that's an example of an intoxication. So intoxications are always going to have a more rapid onset than an infection. A few more things. We talk a lot when we talk about foodborne illnesses. We talk about fecal-oral transmission. It's how most of these foodborne illnesses occur. And it can come from water that gets contaminated, whether it's by runoff from cattle, whether it's by human waste, produce that's washed in contaminated water that's not cooked, or after it's been washed. It can happen from a lack of hand washing. You can have food preparers who don't wash their hands afterwards. It can happen during slaughter. So if during the processing time with a meat, if the gut's nicked and those fecal bacteria can get into other parts, that contamination can happen. But what's important is when you discuss the fecal-oral transmission, if you're ever asked to give examples of it, how it can happen. One thing that's important is you need to address that the food itself will be ingested, that the water, people are going to drink the water. So, if you say a restaurant worker goes to the bathroom and washes their hands and then they handle the food, well, that gets the food contaminated, but you got to get it into the next host. So just make sure when you address that you complete that cycle. So the water might be contaminated, but the pathogens are not going to be problematic until someone actually drinks that water. So just make sure that you complete that whole process, because these guys have to get from a portal of exit into a portal of entry. And so they have to do that in order to cause disease. Now, we spend most of the unit on bacteria. We talk a little bit towards the end about viruses. And, I know that you all this semester of the class probably know more in general about viruses than any class that I've ever had before. But we know that viruses are not living organisms. They're nucleic acid and protein. They have to have a host. So viruses they don't have all the machinery to do replication on their own. They can't do that. So what they have to do is they have to basically hijack a host cell and then have the host take over and do that. And essentially the host turns into a virus-making factory. Norovirus is a very common foodborne illness. That's a virus. He's kind of the cruise ship guy. You hear about him a lot when people get really sick on cruise ships. Prions, we hit on those just a little bit. Prions are also not living organisms. They are just simply proteins. And they typically are important in cell-to-cell communication, but when they can get misshapen. When they get misshapen, they can just wreak havoc, and they can also cause other prions to become misshapen as well. And if you're familiar, and which you should be if you've followed through with unit or with section 4 of unit 1, but when we talk about BSE, the bovine spongiform encephalopathy, which is what you've heard of as mad cow disease. That is that's when it happens in the cattle, and that's a prion disease. And then we talk about Creutzfeldt-Jakob disease, which happens when it occurs in a human. So that's the human variant of it. So we cover bacteria, which are prokaryotes, viruses and prions, which are non-living, And then we also talk about eukaryotes. Eukaryotes are going to be your more complex living organisms. They're multicellular, like I said, more complex than the bacteria. We talk about tapeworms, protozoa. When we talk about tapeworms, again, these guys are going to be much more complex. Their cells are going to be more complex. Their life cycles are going to be more complex. One thing with the tapeworms that we talk about, or the helminths that we talk about, is that they can form cysts. When they form cysts, and don't think of it like cysts. If a lot of times we use the word cyst, you'll think about like, oh, if you have a cyst on your body. This is different. What these guys are is they're just these resting non-modal states where where their transmission can occur. They're important for disease transmission. So what will happen is these helminths will form a cyst, and it's part of their life cycle, and it will allow them to withstand unfavorable environments, and it will allow them to get from one host to another. So those are very important for disease transmission. Again, it helps them get from a portal of exit to a portal of entry, or to go from host to host. So that's very important for them. But for, like I said, for Unit 1, I know we cover a lot. There's a lot to take in. But as you're going back through it, if you have any questions, let me know. Like I said, this was just kind of a little bit of a high point of when I was grading, when I was looking back over things. I kind of just took some notes about things that, maybe could use a little bit more clarification for everyone. So I hope that this is helpful. And again, as you're studying, if you have any questions, any concerns, please don't hesitate to let me know. I'm here to help. And thanks for your time. Take care.